Welcome all. Uh, so we will start with the paper 1 of semester 3, microbial physiology and metabolism. So this paper deal with all types of the metabolic cycles that occurs in the bacteria and the physiology of the bacteria. So the physiology, when it concerns, it mostly about the growth of a bacteria and the things are required for the growth of a bacteria. So the growth is actually a very physiological mean aspects of the bacteria that ultimately leads to the metabolism and the energy generation. So throughout this we will learn different aspects of metabolism and the physiology. So unit 1 is mostly concerned with the bacterial growth. The growth is what we all know. It is the certain changes in the bacteria that leads to the doubling of the cells. So when we have to define a growth, it is actually the orderly increase in the quantity or the structure of a cell. That means when a small cell is there, it gets bigger, but in the increase in the their mass of the structure. That means that every cell components they get duplicated, so that the ultimately cell get reproduced. So reproduction or cell division is mostly by the asexual process. That means the bacteria, they always reproduce by the binary fission. That is the reproduction process for the bacteria. So what we going to learn in this, it is what the first is the definition of the growth. What is that? It is the orderly increase in the quantity or the structure of a cell. It's very simple. A small cell get bigger, so that means the mass is getting increased. And then second is what? The bacteria divides by the asexual way of process. That is the binary fission. So during the growth of a bacteria, these 7 developments generally occur that ultimately leads to the suppression of a cell. That means the reproduction process. So whenever we keep the bacteria into the medium, so that medium is actually transferred its nutrients into the cell. Second is that nutrient, it converts into different materials that is required for the growth of a cell. That is the protoplasmic materials. When these two things generally happens, there is the increasing amount of the nuclear material. Nuclear material is what? Now it's DNA and the RNA. So the mostly all the molecular biology process regarding replication, transcription and the translation of the DNA generally occurs into number 3 stage. Okay. Once the DNA gets replicates, all the process happens. The cell, it gets elongates. Elongates means they become a little bit pull apart. Okay. So during the cell elongation, what happens generally? There is the invagination. That is a small kink is there between the cytoplasmic material. Okay, and that leads to the formation of the cell wall. So what generally happens? Nutrients enters into the cell, creates a certain essential materials of the bacterial cell. Then DNA get duplicates. After that, cell become longer, and then there is the formation of the cell wall. Okay. Finally, the daughter cells, that the new cell that is produced, it is producing from the old one. Okay. So when there is a formation of a cell wall, that means the cell constrict and duplicates into the two. That is the ultimately leads to the separation of a daughter cell. So this is what the process of replication of a cell, particularly that is called, we call it binary fission. Okay. So what happens in the binary fission, as I told you, when we keep the cell into the medium, the nutrients from the medium enters into the cell. They started producing their different cellular components. Okay. During that phase, mostly in a cell division, the phase S, that is a synthetic phase, where the DNA is actually get synthesized. Okay. So these DNA synthesized, get duplicated, and then it produces certain process like transcription and the translation. So the all essential necessary proteins are synthesized. Once this all happens, the cell enters into the second phase where the cell wall on the plasma membrane they created, and then what, they pinch inward little bit like this. Okay, this is called a constrict. Cell wall and plasma membrane they pinch inward. Okay, and when the pinching inward is too intense, there is a formation of a wall between these two, that is the cell wall formed between these two cells. Okay, so during happening this, the copy of a DNA is also getting duplicated and transported into the each daughter cells, and finally the pinching is so intense that separates the two cells like this and like this. Okay, so this is overall what we call is a binary fission. Now this binary fission is actually a way of duplicating this cell for the bacteria. Now this process of asexual reproduction mostly occurs in all types of bacteria, where a single cell converts into the two cells by means of a asexual reproduction. Okay, so up till now this is all clear. What we have learned into this, it is the growth definition, it's how the cell get divides. During the division, what generally occurs into the cells. This is all 7 steps that ultimately leads to the separation of two daughter cells. So this is up to the binary fission is about the topic for the reproduction of a bacteria in your syllabus. Bacterial growth curve is the new topic, and this is most important topic. Okay? Mostly 10 marks question comes from this bacterial growth curve. So as I told you, the bacteria during the growth, okay, they generally go to certain phases, and these phases is actually explain how exactly the bacteria grow into the numbers. So for this, what we have to do, we have to inoculate a small number of a bacteria into the medium, okay, and then count the number of a bacteria every hours, okay, for the 24 hours continuously. So ultimately what we get when we plot the log number of cells against the time, we obtain a curve. Okay, and the curve is what it is like this. Okay, it mostly consists of 4 phases. First is a lag phase, log phase, stationary phase, and the death phase. So starting from the first phase, that is the lag phase, that means the time from the inoculation of bacteria into the medium. Okay, in fact in this first phase, what happened when we add the bacteria into the medium, okay, not bacteria immediately start its growth, okay, or the duplications. Okay, first of all they prepare themselves for the growth. That means what happens, the cell wall is created, plasma membrane is created, DNA get duplicated, all these things happens into the lag phase. So ultimately what the bacteria, they are preparing for the growth in the first phase. There is no increase in the bacteria, they get ready for the increase. Okay, once this phase completed, all the metals they have been ready, the bacteria now enters into the second phase. Second phase called a log phase, where the bacteria exponentially increase in their number. That means they grow very fast. So why the growth is very fast in this particular phase? Because all the things necessary for the growth is already produced into the first phase. So the bacteria start binary fission, they start division rapidly into this particular second phase. That means the log phase, where the growth is exponential, that is the increase in the number of bacteria is much more. So once the second phase is finished, there is a straight line obtained. This is called the third phase, that is the stationary phase. What happens in this, there is the equal number of a bacterial cell living and equal number of bacterial cell are in the death. Okay, that is the death phase. So there is a counterbalance of the living cells and the dead cell in this particular phase. That is the curve is most likely in a straight line, that is called the stationary phase. There is a balance between the living cells and the dead cells. Why there is a dead cell? Because the sum of the medium, that is the nutrient medium we provide for the growth of a bacteria, is depleting, that is it's a decreasing, because all the growing bacteria, they utilize the medium and grow. So the number of bacteria grow, that means there is a less amount of nutrition available for the other bacteria. So the bacteria, they start to die, and whatever the nutrient medium they are left will be utilized by the bacteria, and then they grow. So there is the two types of particular things happen. Some bacteria, they are growing, and some bacteria, they are in the dead phase, they are dying because there is no nutrient is depleting. Okay. So once this phase is occurred, finally it comes into the fourth phase. Fourth phase, what is actually a death phase. Okay, there is the exponential decrease in the number of the living cells. Why there is a dead cell? Because there is no available nutrient remain in the media. Okay, all of the nutrient media utilized here in the third phase, here in the second phase. Okay, so all the nutrient media utilized by the bacteria, finally there is no nutrients in the mediums, so there is no nutrient, there is no growth of a bacteria. Ultimately the bacteria, they go in the death phase. So the bacteria die particular time into the phase. Okay, so this was, there are the four main phases. There are actually 8 phases, but that is not so important. So these 4 phases are very important: lag phase, log phase, stationary, and the dead phase. Okay. So what we have learned up till now from these 4 phases of a bacteria, we add the bacteria into the medium. That is the first phase, that is the log phase, where the bacteria utilize the nutrient media and synthesize different cellular components like cell wall, cell membrane, then DNA gets synthesized, replicates, and ultimately bacterial cells are ready for the duplications or the increase in the number. Okay, so in the second phase, they start growing exponentially. The number of the bacteria get increased. That's why the curve you see, it is a quite rise up. And the third phase, where there is the equal number of the bacteria living and the dead. Okay, some bacteria, they still living, some bacteria are in the dead phase because they do not get any nutrient. And finally in the fourth phase, as last 2 phases utilize all the nutrient, there is no nutrient remain in the medium, so bacteria won't get survive. So there is the declines in the number of bacteria. So this is what the 4th phase of the bacterial growth. Here you can see this. This is the first phase, second, third, and the fourth phases. All these 4 phases. This is what a new inoculating bacteria in the medium, where the log phase, there is no growth, but bacteria, they are ready to grow. So there are so many, like very few cells. When the log phase is started, the bacteria grow very exponentially. Okay, all these living cells are there. In the stationary phase, the living cells are still there because nutrition is still there, but there are certain bacteria, they also undergo death. Okay, very small number, but so there is a counterbalance between the life and the death in this stationary phase. And then finally, when all the nutrient is completely exhausted, there is what? No viable cells remain, so the dead cells are more here. So this is overall the growth curve pattern of the bacteria. So what we have learned from this, it is what? The bacteria, during its growth, they undergo 4 type of phases, and these 4 type of phases very well explain the mechanism of growth of a bacterium. You have to remember the 4 phases of this: lag phase, log phase, stationary, and the death phase, and what happened into each of the particular things. You have to remember this. Okay, so once is what, in case of these 2 phases, stationary and the death phase, during the growth of a bacteria and due to the depletion of the medium, okay, these are the 2 things are responsible for the death of a bacteria in the last phase. Okay, what happens as there is no medium? First thing. Second, there is the accumulation of toxic waste, and because of the toxic waste, bacteria won't survive. So these are the two reasons why the bacteria die into the particular their life cycle. One is there is no nutrition, and second there is the accumulation of toxic waste. These two things, right? So this is overall your bacterial growth curve. Remember, you have to remember what it is, only the 4 phases, right? Hope this is all clear up till now. Now the next topic is what is the generation time. Okay. So what generally generation time is what? It is the time required for the cell to divide. Okay. Or to get double. That means जैसे हमने देखा था from the lag phase, when it goes into the log phase, the number of bacteria increase. That means there is something, a time that is allow the bacteria to divide, so to get to double. That is called the generation time. Okay. So bacteria, we know it is reproduced by the binary fission. Okay. So from one cell it becomes 2 cells. So we can represent like this: one cell converts into 2 cells, that is the binary fission, 2 to 4, 4 to 8, and the so on up to the 2 raised to n. Okay. So what is generation time? It is for the E. coli 23 minutes. Means during the 23 minutes E. coli cell get double. Can you understand this? Yeah. As aureus require 15 minutes to get double, and tuberculosis more around 900 minutes to get double. Okay. So this is what 23 minutes, 15 minutes, 900 minutes. These are what? These are the generation time. And what is generation time? Generation time is the time to required to divide or to get double, right? So this is the generation time. And now there is an expression of a growth. Okay, what is the how the growth and what number of a particular growth can be produced during the 24 hours, we can mathematically represent. This is one of the very important topic, mostly asked for the 2.5 marks. Okay. So what we need to do in this case, we have to calculate the generation time, that is how many times it required to get double of an individual microorganism following the experimental data. Okay. Now which experimental data we needed? Number 1, we need the number of bacteria present at 0 time, that is mean at the start. Second, number of organisms present at the end of time t, and the third is the time t, that is time interval. All these 3 things we are required to calculate the generation time of a bacterium. As I said here, generation time for E. coli is 23 minutes. That means it is calculated, right? So we need to see how we can calculate the generation time of any bacteria. For this, what we have to do, we have to check out for the one expression, that is B is equal to 1 is to 2 raised to n. Okay, we add the cells into the medium and add given particular time period, we have to calculate that. Okay, how to calculate? We have to express this like B is equal to 1 is to 2 raised to n, where 2 raised to n is the bacterial population after n generation. Now n generation means what? The time required for the population to get double, right? But what happens under practical condition? Take the bacteria, we won't add into the medium. Okay, we take what we do in during the inoculation, we take insert the needles, okay, into the agar saline and then put into the broth culture. So when we take the bacteria into the inoculating needle, there must not be a single bacteria. Okay, there are thousands bacteria which are present on the loop of the inoculating needle. So what we need to add this bacteria into the medium. So practical is what we are adding, actually thousands of bacteria into the medium at 0 time. So the formula of now here it become different. It is the Bn is equal to B0 into 2 raised to n. Okay, what are these? B0 is number of organism at 0 time. Bn is what? Number of organism at the end of n generation, and n is equal to number of generation. Okay, yani ke what we have to understand here, thousands of bacteria reproducing at 0 time, not a one bacteria. So in this case we have to change the equations. So what we do, this is the equation number 1. By taking the log of equation number 1, so it become log bn is equal to log b0 plus n log 2. Okay, so we can reshuffle this n log 2 as n log 2 is equal to log bn minus log b0 exactly. Now calculating for this n, so it become bn minus b0 divided by log 2. This is what is simply reshuffling of the terms. Okay, it's very easy mathematics, not that too much tough. Thus we can calculate the number of generation when we know the initial population b0 and the population at the particular number of generation after time t. Okay, so n is what is the number of generation, that is how many number of generation it requires. Okay, the generation time g is equal to the time divided by number of generation, that is g is equal to t by n. Okay, so n value is this, we know this. Okay, what we do, we'll put this n value of this here. Okay, so n is what, log bn minus log b0 divided by log 2. So we simply put this value of n here, exactly here. So g is equal to t यहाँ पे है, divided by n की वैल्यू क्या है, it is this, right? So finally simplifying or calculating for this g, what it become, g, that is means number of generation, growth rate, t is t log 2 divided by log bn minus log 0. So this is the mathematical expression of the growth, that is the g, right? So this up till now we have understand particular this. Okay, mathematical expression we have to calculate or we have to derive it. Okay, why we need to do, we need to know the time that is required for the bacteria to get double. Okay, so simply we have to see this equation 1. Okay, thousands of bacteria we are adding into the medium, then this equation it comes here. We have to simplify this equation just into this, like very simple logarithmic reshuffling of the terms. And then generation time ka there is a formula, g is equal to t by n. So we have to simply put this n value of this here, and finally we get this g is equal to t log 2 log bn minus log b0. This is the mathematical expression of the growth. Hope you all understand this up till now, right? So when we recall this, okay, summary of this class is what we have learned is the definition of a growth, how the bacteria they duplicates or they replicates. Okay, this is by the binary fission method, where the single cell converts into two different daughter cells. Then we have seen the growth curve, which is very important topic. Okay, growth curve has a 4 phases: lag phase, log phase, stationary phase and the death phase. Okay, जहाँ पे log phase all the bacteria become ready for the growth. In log phase there is a increase in the number of the bacterial growth. Stationary phase there is a balance between the living cells and the dead cells, and the finally death phase where all the bacteria they are undergo dying process. Okay, and this is the generation time. Generation time is the time required for the cell to divide or to double, right? So this is 23 minutes is the generation time for the E. coli, 15 is for S. aureus, 900 for the tuberculosis. And this is what we have expressed the mathematical derivation for the growth. Okay, where you have to simply focus on this particular equation. Okay, and then simplifying this equation like this, it is very easy. Okay, and therein it comes the value of n. Now this value of n you have to simply put in this particular equation, g is equal to t by n. So put this n value of this here, log bn minus b0 divided by log 2, right? So this ultimately become, after reshuffling, g is equal to t log 2 by bn minus b0. So this is overall for the today's class. Okay, the first topic of this semester 3 paper 1, that is mostly concerned with the growth of a bacterium. Okay, so in the next couple of days I will put you the another class for the same semester. Up till now, um, keep on looking into this particular video. Okay, mostly use the earphone so that the voice would be very clear. Okay, after a few time we will have I will provide you the notes on this. You all people have to log in into the particular TeachMeet app. Okay, most of the people they have joined, but some of are still yet to join. There itself I will provide you the notes of these lectures. Okay, thank you. Keep studying. Stay remain healthy. Thank you.